Okay, so welcome to this tutorial, the physiology tutorial, an introduction to excitable tissues. Actually, a very much important topic um, in physiology. Okay, so my name's Excitable. I display there the physiology tutor, so teaching the Step Up Academy. Okay, so today's session is actually very simple. Just want your attention as we go through it. It's actually a relatable topic and very simple to understand. Okay, so first of all, you need to understand what do they mean by excitable tissues? What does the statement excitable tissues mean? So excitable tissues, they are actually those tissues which are able to propagate an action potential. An action potential is uh, more like a nerve impulse. Okay. So in our daily life, we tend to, we're able to move objects, we're able to speak. Even right now, as you are getting me, it's all about action potential because the action potential is generated and then the sound is transmitted like that, sent to the brain, then the brain interpret, then it's sent back, then you're hearing my voice, even though it happens within seconds. But that's all about excitable tissues. So excitable tissues are tissues which are able to propagate an action potential. Okay, so we basically have four types of tissues. Among them, we have the nerve tissue, we have the muscle tissue, epithelium, and the connective tissue. Now, the one I've put in both, there is nerve and muscle. These two are considered to be special. Why? Because they're excitable tissue. They're able to generate an action potential. Okay, so we'll start with a nerve tissue, then we will discuss a muscle tissue in the next session, maybe even after the following session. Okay, so basically, the neurons are the basic building blocks of the nervous system. So the human central nervous system comprises about 100 billion neurons and 1.5 trillion cells. Okay, so you know to say that the neurons are the ones which make the nervous system. So it means that they are interconnected, connected together for them to form the nervous system. So the nerve tissues are the ones which combine to form this nervous system. Okay, so this is a typical structure of a neuron. So a neuron has parts, and these parts play a very important functions. Okay, so when you look at the neuron, we have the cell body, which contains the nucleus. Then we also have the dendrites, we have the axon, we have the myelin sheath, we have the squamous cells, we have the node of Lange, and we have the axon terminus. So I want you to look at the structure of dendrites. Okay, they are looking more like receptor. So these dendrites receive signal from the neighboring neurons or neighboring cells. So they receive signal and electrical impulse in form of an action potential. Okay. Then the function of the nucleus here is just to control the activities within a neuron. Okay. Then we have the axon terminals here. These axon terminals are the nerve end. So mostly they transmit a nerve impulse from one neuron to another or from a neuron to a muscle, any effector organ, I can say. Okay. Then there's very much important point to note here. We need to know that the node of Lanvia mostly contains a higher concentration of sodium, even though when you look at the axon terminal here, here where there is an arrow which is pointing here, there's what we call the axon hierarchy. There are some theories that axon hierarchy is involved in the propagation of action potential. So those are just important points to note as we are um, be looking at in this chapter. Okay. Then this structure will act as our reference in this session. Okay. So actually the nerves are classified. They are classified in different ways. So we are going to look at the classification of nerves depending on various ways. So the first classification of nerve is based on the function. We have the sensory nerves or the afferent nerves and the motor nerves, the efferent nerves. So the sensory nerves, their function is just to sense, to detect what is in the environment. Then the motor nerves, they just send the nerve impulse to the effector, more like the muscle for you to react. So let's imagine something like uh, there's a source of heat, or maybe say a needle. You prick yourself. What are the nerves that are going to be involved there? The first ones, they are going to be sensory nerve to sense what is happening. There is a prick. So at that point, information will be translated, will be transmitted to the central nervous system, which is the brain, and later on return via the motor nerve for it now to be to be felt. Okay. So the effector organs like the muscle, it need to be sent back for you to feel it. So that's a function based on the function. So the sensory nerve sense what is in the environment, and the motor nerve, they bring back the results to you. Okay. That's why when you prick yourself, you feel pain. It happens within a short period of time, but the body would have sent that information to the brain for it to be transmitted. Okay. Then we also have a nerves classification based on structure. We have the myelinated nerves and non-myelinated nerves. So the myelinated nerves, the major difference between these two is based on the structure. So the structure is just the myelin sheet. So I said this is going to act as a reference. So we have this one cell. So the myelin shield. So the myelinated ones have myelin shield on in their squam cells. Okay. So these squam cells, they have myelin sheet. But the other one, the non-myelinated one, they don't have squam cells in there. They don't have myelin in their squam cells. So that's what differentiates a non-myelinated nerve from a non-myelinated nerve. And this myelination is very much important because it's also help in the fast transmission of electrical potential and protecting the nerve from any trauma. Okay. Then we also have based on distribution. Based on distribution, we have the somatic nerves and the autonomic nerves. So the somatic nerve, usually they innervate something that you can control. Okay. So like skeletal muscle. You are there, you are in charge of what is in the environment. You are there to make a decision. Now I can lift a bottle. Now I can let go of a bottle. But for all areas of parts that are innervated by autonomic nervous system, they do things by themselves. So the best example for autonomic, the organ that's supplied by the autonomic nerve is actually the what's this? The heart. So the heart pumps blood on its own. You don't tell it, even when you're sleeping, it pumps on its own. So that's what's based on distribution. Then based on origin, origin is just about where is this nerve coming from. So the nerve can originate from the brain, we call it the cranial nerves. Then originates from the spinal cord, we call them spinal nerves. Okay. That's simple as this. Then we also have classification based on the conduction velocity. The conduction velocity is just about how slow or fast this nerve is able to conduct an electrical impulse or action potential. So we have type A, which are actually myelinated. Under type A, we have alpha, beta, gamma, delta, and gamma. So these type A, they are usually larger and they are myelinated. So they transmit nerve impulse at a faster rate than any other type based on the conduction velocity. Then we have type B, which is also myelinated, but they are quite smaller. They are in a medium size as compared to type A. Then the last one we have type C. It's actually the smallest and it is non-myelinated. Now I explained the importance of myelination in a neuron. So these the swan cells, they are actually myelinated. So once they are myelinated, I say that's very much important in the conduction of faster conduction of nerve impulse. Then apart from that, I also say that it protects the nerve from trauma or anything. Okay, okay. So without wasting time, we are going to axonal transport. So axonal transport is just about how a nerve transport electrical impulse from one nerve to another. So the movement of electrical signal from one neuron to another neuron, it is usually done through microtubules. Okay, the microtubules are part of the cytoskeleton. We have the microfilament, microtubules, and the intermediate ones. But the microtubules are there, which are one and transmit the electrical signal from one neuron to another. Then the classification depends. Okay, we have the anterograde and the retrograde. So the anterograde is when a nerve impulse is moving from, let's go to our reference, from, it's moving actually from the cell body towards the axonal terminal, the normal one. So the anterograde is when it is moving from this area, then it's going, let me just highlight there. Okay, so anterograde is when it's moving from this area, going this side. Okay, this is the normal movement of the, the electrical impulse. It needs to move from the dendrites going to the axon terminals. However, there are some instances when it moves backwards. It moves from the axon terminal coming toward the dendrite. This we call the retro. Okay, the retrograde. So let's hope everyone is clear there. So we went on to the, yes, that was the movement, the type. Yeah. Now, there is a very much important uh, point that I need to mention. We are just looking at the neuron, the structure. Now, we need to look, we've been talking about an action potential, we've been talking about electrical transmission. Now, we need to look about how an action potential is actually generated, because it's very much important for you to understand how an action potential is generated in the human body. Okay, so mainly the genesis of membrane potential, this is the process of how an action potential is generated, which involve the resting potential, the resting membrane potential, then going to polarization, then going to depolarization, going to repolarization, and eventually going to hyperpolarization. Okay. So if you have a paper right now, you can actually write this down so that when I go on the diagram, you can relate when I'm explaining. Okay. So we actually have this diagram. So what really happens? How does an action potential, how is it generated? That's what we're interested in here. Okay, now in the introduction to physiology, you learn the functional morphology of a cell. We looked at the extracellular fluid and intracellular fluid separated by a cell membrane. Okay, the cell membrane is the one which separates the extracellular fluid from intracellular fluid. And then there are some specific ions which are actually inside the cell and some specific ions which are actually outside the cell. But the major principle is to say that for these liquid to be at equilibrium, the cation intracellularly and the anion need to be equal to the cation and the anions outside the cell. So at this point, mostly the cell is usually at rest. So I mentioned today that resting membrane potential. What do we mean by resting membrane potential? Resting membrane potential, let's look at this green line here. So this green line will act as a cell membrane. Then we'll assume that this is the ECF. ECF. Then inside, we'll look at it as the ICF, meaning intercellular fluid. Okay. So mostly, potassium is more inside the cell, and sodium is more outside the cell. And sodium plays a very crucial role in the generation of an action potential. Okay. Then outside is more positive as compared to inside. Inside is more negative at this point. But what happens is that there are what we call leak channels on the cell membrane. So these leak channels, what do they do? These leak channels, they allow slow influx of sodium ions to enter inside the cell. The leak channels are not big enough to create the potential difference here between outside and inside. Inside is more negative, outside is more positive. At this point, we call, whenever there's no activity that's happening in the intracellular fluid and extracellular fluid, we call this resting membrane potential, meaning that there's nothing happening. And as I said, what's happening here is that we have leak channels, more like leak channels. These leak channels allows slow inflow of sodium channels. So once sodium start entering inside the cell, it starts creating a potential difference. At that point, we call polarization. Polarization is when the potential difference is start to change, okay, from a certain millivolts to a certain millivolts. It's no longer at resting membrane potential. So at this point, what really happens is that sodium start entering. Once sodium start entering, it's making inside more positive. Initially, I said it's negative. It's making it more positive because sodium is actually a cation. It's a positively charged. So inside, the resting membrane potential is about, in a neuron is about negative 70 millivolts, as is stated there. But since sodium starts rushing inside the cell, it starts to change until it goes somewhere to about negative 55 because sodium is entering and it's positive, but it's changing the membrane potential from -7, the resting membrane potential from -7 to somewhere negative 55. But what really happens physiologically at negative 55, there is what we call opening of sodium gated voltage gated sodium channels. The voltage gated sodium channels, they are more porous. They allow sodium to rush inside the cell more than what the leak channels were allowing it to enter. So the voltage gated channels actually plays a role in the fast influx of sodium inside the cell. Okay, so influx is just about the movement of sodium rushing inside the cell. Then reducing at this point, since it was -5 and sodium has exceeded the limit, once it reaches a -55, we call this a threshold. At this threshold,